I'm very glad that we have Alex Gramfort in Brainhack Donostia as one of our keynote speakers. So Alex is a senior researcher in India, working in the parietal team in Paris. He was formerly an assistant professor in Telecom Paris Tech, one of the most prestigious engineering schools in France, in the image and signal processing department from 2012 and 2017. He's also affiliated with Neurospin. His field of expertise is very broad. So he's an expert in signal and image processing, in statistical signal processing, in machine learning, in deep learning, as you will see afterwards, in scientific computing, in convex optimization, in optimization problems, and mainly uses all these tools to understand how the brain works in using functional neuroimaging data, either EEG, MEG, or functional MRI. He collaborates with many people, interfacing in a lot of fields from physics, computer science, software engineer, or neuroscience. He has co-authored many papers, even more conference proceedings since 2009. And he's usually invited to many conferences to present his, as a keynote lecture, his latest developments. He's also very committed to open source software development. He's a core developer of the scikit-learn machine learning package in Python, which is, I would say, the most important package for one of the most important packages for data analysis in Python. He's also the leader of M&I Python software, and that is used to analyze, as you have seen, in EEG and MEG, and is used across the entire world. He was awarded a very prestigious research grant by the European Research Council, a starting grant in 2015 in a project called Signal and Learning Applied to Brain Data. And in 2019, he was awarded a grant, a very prestigious grant from France called Brain. Personally, I would say that working in signal processing, I'm always enthusiastic and reading very deeply all his works, all the work that he is producing, because he gives and fosters and promotes the new tools and new ways and new ideas. So I am looking forward to your talk, Alex, and the floor is yours.
Thank you very much for the very kind introduction. So I will put the link to my slides that you can download in the chat. So if you want to follow, and if you want to go if you want to go faster, and because I'm too slow, you can also do this. And let me now probably share my screen. All right, so kind of the mission that I assigned to myself for this this talk is to be a bit tutorial style, because I don't know exactly what's the background to all of you, but I think it's probably a good use of this presentation time to also shows you the work of others, and also hopefully introduce a number of concepts and prevent mistakes in your data analysis. So I will take probably 40-45 minutes if it's okay, and I will answer questions afterwards. All right. First, I will start by basically shamelessly advertise some review works that we've published over the last few years. This is a book chapter that you can find freely online, where we discuss what is machine learning to decode and encode neural dynamics, with many co-authors. A lot of them are actually the people in the M&E Python software who wrote this MNE decoding module that you can use to do machine learning using the MNE Python software. And another one, which is another review that we've been putting together with colleagues also in Montreal, where we've tried to review what are the recent use cases for deep learning applied to EEG. And this is something that also comes with a website where we can try to keep up to date what people are doing using deep learning on EEG. And this is a good insightful paper to also know what are the trending topics in deep learning for EEG. All right, so let's step back and look a bit at the big picture of machine learning and EEG and MEG. And first, I will start by basically splitting the world of machine learning in two, even though, I mean, I could be more specific than this, but I would say for this talk, two is enough. So the first part is what people call unsupervised learning, which is what you see here. And this is useful for many things. For example, we use it a lot for data exploration using EEG. This is what people do with ICA, for example. I will mention briefly afterwards convolutional sparse coding. People also use PCA for denoising. All these techniques are machine learning techniques, multivariate techniques that are unsupervised. And on the other side of this slide, you have the supervised techniques coming with different applications like brain-computer interfaces, sleep staging, like you will do in the hands-on afterwards, where you will try to reconstruct hypnograms from EEG recordings. You may want to have also find biomarkers or try to predict what the subject is doing in the machine. All these techniques can be seen as supervised techniques in machine learning sense, and they have many techniques to do this. You know probably about LDA, logistic regression, support vector machine, common spatial filters, convolutional neural networks, which you will be using in the hands-on. And I will also briefly mention these Riemannian techniques that have now been very efficient applied to EEG and MEG over the last 10 years. All right, so the outline of this talk is the following. I will first show you a few papers that basically I read over the years and for me that were quite inspiring. Then I will go again about what is evoked activity and induced activity and insist on the fact that these techniques require different tools when it goes to when you do machine learning. And maybe explain a bit what questions we may want to answer using machine learning. And then I'll try to use this time to introduce some algorithms going from linear models. That would be a good start to go towards deep learning afterwards, and then insist on good practices and how to do properly cross-validation and reporting your results. All right. So let's start with these inspiring examples. So first, this is a fairly old paper now by the people in Aalto in Finland, where they've done this experiment where people could see these types of visual stimuli which are oriented gradings with two different frequencies. Here, one frequency, low frequency, high frequency, and two different types of also orientations. And what the machine learning algorithm was trying to do is that taking the data at every millisecond, trying to predict which of the categories of stimuli was it. So which spatial frequency and which orientation. And you can see, for example, at what time instant do you start to see an effect in the brain. And this is now quantified using a cross-validation score using a decoder. Okay, and they could claim, for example, that here, as early as 50 millisecond post-stimulus, you already have something happening in the brain that is enough to decode either spatial frequencies or orientations. So this is related to time-by-time decoding, and here I give you a link at the bottom to do these types of analysis using the MNE software. Another example is related to this one. This is what has been now called time generalization. This was pioneered during the PhD work of Jean-Amy King, at least in the context of MEG and EEG. And the idea is that you will learn on a given time instant and trying to predict a target variable at a different time instance. And for example, looking at these pictures, which are basically what are generalization matrices, I will come back to this later in the talk. You can see if the activity in the brain is sustained, like here, or if it changes, if the polarity is inverted, and basically test different hypotheses about how the brain is responding. Is it a sequence? Is it sustained? And things like this. This is what is called generalization estimator in the M&E software. Another work, which was, I think, quite also inspiring in the community, was this work by Radek Kishi now 6 years ago, where he presented many objects using a technique called RSA. He could test basically at what time instant could you differentiate, for example, between animate or inanimate objects. And here, this is the MDS, so the 2D projections of the stimuli. And here, using a different types of condition comparison, using here natural objects or artificial objects, and seeing again the when, which is when is this information encoded. And this was made possible by using MEG. So Radek Kishi shared with us now a few years ago one subject from his study that you can basically explore and play with using this decoding RSA example, and you have the link at the bottom of this slide. And another types of approach, which is here related to encoding techniques, has been quite popular in the groups are interested into speech processing and how the brain processes speech. So you have an input speech stimulus, and what the machine learning technique is learning how to predict the brain response. So giving us here a spectrogram that you see here, can you predict the brain activity? And these are related to techniques what I call here ST, which stands for spatial temporal receptive fields, or STERF. And if you go to this example, it will show you examples. Plus, if you go to this webpage, you see an example of how do you do these types of analysis using the M&E software. Enough of supervised techniques, now illustrating some unsupervised techniques. So this is a slide that I took from the group of Scott McKeague at UCSD. Very popular team who have pushed the limits of ICA over the years. So this is raw EEG data. This is an example of ICA decomposition, where you see the EEG artifacts that are popping up. You see a very nice alpha burst. You see here EMG artifacts. You see the ECG. So ICA completely in an unsupervised way without any human supervision, just using independence assumption, is able to unmix and uncover these types of sources from the multivariate EEG data. Something that we've been working on recently in my group is called convolutional sparse coding. You can see this as a variant of ICA, where we're trying to decompose the data into components, which are here topographies. And then the time course, rather than being based on independence, we assume that the time course is produced by a prototypical waveform, and here an activation pattern. So basically, you are saying that the data is a topography, and then this waveform is replicated at this instant, this instant, and this instant, and this is how we decompose the data. And by using this convolutional sparse coding, what we are able to reveal is we can learn the temporal waveform. There's been a lot of activity about basically waveform learning and how the variability of waveforms can be informative. And this is a tool to start to address this question. All right, so let me now step back and maybe try to explain why these types of techniques works and what are the key ingredients. So the first thing is you need to understand a bit the physics of MEG and EEG to understand why it can work. And even though you can say, okay, the brain has a very fine resolution, with MEG and EEG, we see stuff which are rather spatially coarse. Well, it turns out that the fact that the brain is a convoluted structure is very helpful. And here you see the folding pattern of a brain. And if you have three sources which are here, it turns out that when you move along here the gray matter, it will change slightly the orientation of the source. And it turns out that MEG and EEG are very sensitive to source orientation. So just by moving by a few millimeters, if the orientations change, it can produce a very different pattern on sensors and be therefore decodable from using machine learning. And this is now a more concrete, more complex example. So basically the take-home message is that orientation is here very helpful to disambiguate even very neighboring sources. All right, so now this is a very toyish example. I have two sensors. So imagine you have two sensors. They are all seeing the same source, which I here call S of t, plus some added noise. Provided that the noise is shared, which is quite common if you have an artifact that is basically spread over sensors, you can do something as simple as just subtracting these two sensors. If I just x1 minus x2, and you just do the subtraction between these two terms, well, it turns out that magically you only keep the source. So this is a very, I would say, toy example, but it really shows you that if you have multiple sensors, by doing linear combination of the sensors, you can increase the signal to noise ratio. And this is really at the heart of all these techniques that you apply in why people are doing decoding on MEG, because they can really leverage these linear combination of sensors to improve the signal to noise ratio. And if I were just to repeat really out loud, too often people are doing statistics and work by using single sensors on EEG and MEG. And for me, you're really shooting yourself in the foot, because you have so much SNR gain and quality of data that you can improve if you start to combine sensors using multivariate techniques. So here is another slide that I took from a paper from Stefan Haufer that explains basically the same concepts, that if you start to go multivariate, you can disambiguate here two conditions here. I will just move on quickly because I'm running out of time, I think. All right, so let's step back and look a bit at these induced and evoked activity profiles and see how this impacts machine learning. So imagine this is your raw data. This is roughly 300 milliseconds of MEG data. On average, just a chunk of 300 milliseconds of data. If I start to average, so this is averaging 50 trials or 50 repetitions of the same. Here, this is a median nerve stimulation. It turns out that if you start to average more and more, here averaging up to 400 trials, you start to see these butterflies, okay? This is what people call butterfly plots, where here you see the signals that are roughly zero during pre-stimulus, and then you see this nice activity that corresponds to dipoles and active brain regions. Well, it turns out that if your signal of interest is actually evoked, meaning that it's basically always coming at the same latency after a stimulation, and basically it survives averaging, then you need to use certain machine learning tools. But basically, if your activity is not time locked and it's induced, you will need to use different tools. So here now, this is about induced activity. So here, this is simulation work. So roughly a windowed cosine functions with some added noise. And here what I'm going to do is a DFT, which is a discrete Fourier transform. So for using this time window, I'm going to compute the Fourier transform of this. And this will produce these types of image. People call this time frequency representation of the data. So this is basically every column of this image is the modulus or the squared modulus of a Fourier transform of a given time window. And this produces these time frequency transforms. So what's happening is that if you have this multiple repetitions of here a prototypical brain response, if your signals are completely aligned and time locked by just averaging your here 5 repetitions, you get a very clean signal. Basically, if you have 5 epochs and they are corrupted by noise, and these noise is independent, if you average your 5 epochs, your noise variance is basically divided by 5. Okay, this is statistics 101. So this is why you gain in signal to noise ratio. But now the problem is that if your data is not exactly phase locked, so sometimes here you're at the trough of your oscillation, sometimes you're at the peak, and it basically means that by averaging these 5 repetitions, you're going to kill your signal. And this is why people have come up with these time frequency techniques where you, rather than averaging the time series, you're going to average the time frequency representation and here obtain a clean time frequency map. So basically by using this representation, you have a representation which is invariant to the phase, and this allows you to boost the signal to noise ratio. And this is something that has been historically used in the BCI literature, using here what people have called common spatial patterns. So here, this is motor imagery. So you don't move your body. You just think about moving here your left arm or your right arm. Okay, and so actually left hand and right hand. And these basically thinking about moving your hand will produce some induced activity. And here you have two prototypical topographies that here are represented. And basically, common spatial pattern is a way to build very powerful multivariate techniques to decode these types of induced activity. So it's probably not enough used in the cognitive neuroscience literature, but know that if you look in the BCI world and you want to decode induced activity, this is what you should be using or what you should try. All right, decoding versus encoding. I'm going to show a bit of equations here. All right, so in a word, you could basically see decoding as basically going from the brain data and trying to predict mental representations, so behavior, what is the types of stimulus that the subject was paying attention to in the machine. And encoding is basically the reverse. You're trying to predict the neural recording. So you can try to predict the MEG data. You could also predict fMRI data using functional MRI data. And this is basically the reverse. So now I take this cartoon slide to start introducing some notation which are very standard in the machine learning world. Here in the decoding setup, the X, which will be the input of your machine learning predictive model, will be my MEG and EEG data. And what you may want to do is to predict a target Y related to the stimulation, for example, what was presented on the screen for the subject, or is the subject healthy or not? So again, you will want to learn a function, what is called here, a function f, this is here, that takes input data X and tries to predict the target Y. Okay, and machine learning, in supervised machine learning, is pretty much this. You try to learn a function f that takes X as input and outputs Y. And you can formulate this in a mathematical way by saying that you have some input feature vector. Okay, so here you have P features for a given sample, a given observation, that is Xi. Xi is associated with a target, okay, which is here Yi, which is in this case can be a real value or something more complex. And you're trying to learn a predictive function that takes this input feature vector, Xi, and outputs a target variable. And this is what you want to learn. If you do encoding, it's the same machinery in terms of machine learning, except that you swap X and Y. Your input for your machine learning task, it will be your basically stimulus properties like the image or whatever the subject is paying attention to, and you will try to predict the target Y. For example, in the STIRF example I was showing before, X could be the spectrogram of the input speech, and what you're trying to predict is the brain rhythms. All right, let's go deeper into algorithms here. So I'm going to consider, for now, only binary classification problem. So this is what I call classification 101. The target Y that you're trying to predict, given a vector of features of size P, is either going to be -1 and 1. So let's say 1 is if you have a control subject, -1 is you have a patient, or 1 you're looking at a house, -1 you're looking at a face. For the machine learning algorithm, it's just 1 or -1. All right, so let's look at what I call here my first decoder, and just to basically show you that it doesn't have to be complicated. So imagine that your subject attending two types of condition, either using faces or scrambled faces. So two types of stimulus. You have basically two conditions. And the types of question you may have is basically, you want to know, is it possible to predict from MEG data if the subject was looking at a face or a scrambled face? When does this happen? Where is it located in the brain? And this is the types of question you may ask yourself. All right. So let's say you have two sensors, okay? You're using a very cheap system. You only have two EEG sensors. You've spent only $100 to get this. You present to your subject a face or a scrambled face. You record for each of the subjects basically two values. So one value for sensor one, another value for sensor two, and you do here 20 repetitions. That gives you every time a dot. And you want to predict if the subject was attending to either a red stimulus or scrambled face or a face, okay? And the simplest thing you can come up with probably is the following. You're gonna first say, okay, this is 1 and minus 1. And then you're going to compute the mean value of all the red dots, okay? Then you compute the mean value of all the blue dots, which is basically here, imagine that it would be pretty much like looking at the evoked activity, just averaging repetitions of this. And then you can do as a classification strategy, predict what is here called the neurocentroid. So it means that for a given observation, you're going to predict red if your observation is closer to the red dot. And if it's closer to the blue centroid, you're going to predict blue, okay? And in terms of decision function, it means that you separate the space in two regions. One region that is going to be predicted as red, and one region will be predicted as blue. Mathematically, when you do this, you are basically computing what is called here a linear model. You can write this decision function in the following way. Separating the space in two regions separated by a straight hyperplane, which is here a line in 2 dimensions, is nothing more than computing the sign of a quantity, which is a linear combination of your entries in your feature vector plus some intercept, which is called beta 0. So xi are my feature vectors, beta is my linear coefficients of my linear models, and beta 0 is a bias term or interceptor. Okay, so this separation of the two spaces is nothing more than a function that can be expressed like this. And once you've learned your function, and which means that here learning the function is learn the beta and beta 0, you can evaluate its performance by looking on new data, unseen data, how it behaves. And count the number of errors, and it will tell you how good or how bad you are. Now, if you look at another technique called logistic regression, which is grounded on the linear discriminant analysis, which is the equivalent generative models of logistic regression, but let's just stick to logistic regression here. You're going to postulate, okay, this is the strategy of logistic regression. You postulate that the log of the probability of being 1 divided by the probability of being -1 for a given observation xi is a linear function, okay? So you postulate this, and then you use maximum likelihood. And maximum likelihood boils down to the minimization of here a function, which is 1 over n, the sum of n terms. And these terms are here what people will call the loss function. Okay, so what it means to learn here is trying to minimize this loss function, trying to find the beta and beta 0, such that you maximize the likelihood, or here you minimize the negative log likelihood. And minimizing this quantity, which is again minimizing the negative log likelihood, will be your inference, your statistical learning strategy. And basically, when you do deep learning, it's you replace this term by something which is not linear but potentially non-linear by basically stacking logistic building block typically. And you minimize this using an optimization technique or gradient-based techniques. And you hope for the best. And if you have a well-calibrated architecture and you do your optimization and your initialization well, then you can get very good performance. Now, as a remark, in practice, we often also have another term, which is the regularization. So what we're doing is not exactly maximum likelihood, but it's called penalized maximum likelihood. And this is for overfitting reasons. Okay, so basically logistic regression is nothing more than this. And deep learning is just one step beyond this. All right, let's look now at decoding and applying this to decode evoked activity. So for this, you can use this time-by-time decoding. So this time-by-time decoding, here you have an example that you can see here. Here I've got two conditions. Either the subject is listening to an auditory stimuli in his left ear, or a visual flash in the left hemisphere, and you see that the classifier is almost reaching a perfect 100% accuracy at 100 millisecond post-stimulus. Okay, so this is basically looking at the causation score over time. Now you can use this time generalization, where the idea is that you will look at the performance. So this would be time-by-time decoding. And provided that your time-by-time decoding is here good, which means that you're above chance, you have two different types of possibilities. Either it's the same brain region, which is basically consistently sustained and active over this time window, or the other option is that it's just a different brain region, which is as decodable that is happening at a later time instant. And basically, time-by-time decoding is a way to tear apart these two hypotheses to know if it's the same generators in the brain or if it's different. And the way it's going to work is by if the model that you learn at T1 is still performing well at T2, then it suggests that the same generator. And if its performance is dropping when you learn at T1 and you try to predict at T2, then it suggests that it's a different brain generator. So this is what you see on these types of plots. I will not spend too much time on this. You can look at this example to know more and replicate this figure. All right, now let's look a bit at decoding of induced activity. And for this, I need to introduce something that is basically at the core and the core element of the decoding of induced activity, which is the data covariance. So here, if you look at the covariance of here, this is 102 magnetometers that you see here. The covariance is a matrix, which is number of sensors by number of sensors, and it contains over the diagonal the variance of each of the sensors. And on the off diagonal term, this is the covariance, which basically means if you're here blue, and if blue means here negative, it suggests that the two sensors here are anti-correlated, meaning that when one is going up, the other is going down. And if you're red, it suggests that you are positively correlated, which is that one sensor tends to go up, the other sensor also tends to go up. So this quantity is encoded in the covariance matrix that, assuming that your data are centered, which is usually the case because you've high-pass filtered the data, it's nothing more than this quantity here, where T is the number of time points, S would be here your data, with C is the number of channels, and T is the number of time points. This is just a covariance, okay? This is just an empirical covariance. A small remark that estimating this covariance can be tricky when you have a short amount of time, and I invite you to read this work that we wrote now a few years ago with Dennis Engerman that explains how to address this issue. Okay, so common spatial patterns is basically a linear model that takes as input something that is derived from a covariance. So assuming that you take your chunk of data, okay, so you take your epoch where the subject is thinking about moving his left arm or his right hand or his left hand, you compute the covariance of this epoch, and then you find some weight vectors here, Wj. You compute this quantity, which is here a float. This is just a number. This is a matrix. This is a row vector and a column vector. This gives you a float. You pass through a log that gives you a new feature, okay, which is a log power. And it turns out if you take this log power and then you combine them using here a linear regression model, you obtain an output binary classification problem, which basically predicts if you're looking at a right hand, if the subject is thinking about moving his right hand or his left hand. Okay, so common spatial pattern again is something that will you will obtain by you will use in the end by using a linear model on nonlinear features that are derived from the covariance. You can look at these these historical works and this review paper by Benjamin Blanquertz, where he explains how to derive these types of models. I think I don't have too much time to go into the details about how you estimate these filters. I suggest to skip the next slide, which is quite math heavy. And you can look at the PDF of my slides if you're curious about this. So I will just skip this because I think it's just too much. Okay, so now I would like just to advertise some work that I realize that people don't know enough, I think, is this use of Riemannian geometry to decode brain signals. It was something that was really made aware, studied, put on a map and became present in our world in neuroscience and MEG and EEG world. It's the work that was started by Alexandre Barrachand at the GIPSA in Grenoble with Marco Congedo and Christian Jutten. And Alex Barrachand realized that he could use Riemannian geometry to do BCI, and it turned out to bring state-of-the-art performance. And what he realized is that when you manipulate these covariance objects, typically that you use using these common spatial patterns, these covariants, they are basically living in a non-Euclidean world, okay? So here, rather than taking these covariants and seeing these covariants as just a matrix that you can flatten in order to get a vector, you can consider that these objects, these covariants, they are living in a curved world. And one way to understand this is that if you take two covariance objects and you subtract them, you don't necessarily get a covariance, okay? These covariance objects, they are positive matrices. If you subtract positive matrices, you don't get necessarily a positive matrix. So basically the space of covariance is not flat, and it's not a Euclidean world. And once I've said this, it means that you need to change the notion of distance. Rather than using a Euclidean distance, you need to use a dedicated distance for these types of objects. So here, this is the geometric distance that is here illustrated. So this is the matrix log of a matrix, okay, which if this matrix here, this matrix has a number of eigenvectors, so which is here are written lambda k. This norm is the Frobenius norm of this log of the matrix, and it turns out to be just the vector norm of the log of the eigenvalue. And what's funny is that this log, which is coming here, is also the log that was proposed in the CSP literature and happened to be very powerful. But here, this basically it makes this connection. But now this is grounded on the Riemannian structure that you have embedded on these objects. And then once you have this, you can do what I showed before, this Niros centroid technique using these Riemannian techniques. And this is what Alex Barrachand called the minimum distance to the mean. So for a given epoch of data, you compute a covariance, and then you compute the mean covariance for all of your trials for the right hand, you compute the covariance for all of your trials for the left hand. And now, given a new trial, in order to predict if you're thinking about moving your right hand or your left hand, you're computing the distance between your trial and the centroid for the right arm, so the right hand or the left hand. And the smallest distance tells you which class to predict. Okay, so it's nothing more than a Niros centroid after changing the distance and using a Riemannian distance between covariances. And this brings me towards the end of this presentation by insisting on, for me, important take-home messages. So the first one is you should do cross-validation properly. So let me first introduce the two concepts of cross-validation that we have in brain imaging. The first one is called intra-subject. So you learn on session one for your subject, and you predict on the same subject on a different session, okay? This is intra-subject decoding. The other setup is inter-subject decoding, where you want to predict on a subject that you have never seen, which is what you see here. You have here in green the subject that you use for training. So you have n-1 subject for training, and you want to predict for a new left-out subject. Okay, so in both cases, what you need to guarantee is that your data on which you predict are independent left-out samples. But here, in the first setup, you're claiming that your classifier can generalize over time on the same subject, where here, you're claiming that your classifier can generate on a new subject that he has never seen. Now imagine that you have a full data set. How do you proceed? Well, you could do separation between training data and test data, learning your hyperparameters such that they optimize as much as possible the performance on the test data. But what I'm claiming is that if you do this, you're cheating. You're cheating. Why? Because you're using the test data to find the best hyperparameters. For example, finding the best number of neurons in your neural net. And this is bad. What you should be doing is the following. You should need to split your data in three. You have a training data, a validation data on which you, sorry about this. You have a validation data that you will use in order to find the hyperparameters. Okay, so you will optimize your number of neurons or the number of filters or whatever hyperparameters of your model using this validation data, and then only you will generalize to the test data. Okay. Keep this in mind, and then I will show you in a minute why you should care even more. So, advertisements. So you've discovered M&E before, you'll be using M&E afterwards. M&E is really a huge international effort among many universities, funding across different countries in the US, in France, and others. And so I'm really happy to have made this effort so successful over the last 10 years, and now so many people investing their time to collaborate to this project. Conclusion. Why more data is better? So this is a slide that I took from one of my presentations from many years ago, where I was working on fMRI. And what you see here is a very famous data set, the AXB data set from his science paper from 2001, where he showed that you could decode from fMRI objects that were presented to the subjects. So here, this is basically face versus house. And you're using a classifier, for example, a logistic regression or a support vector machine to classify if the subject is watching a house or a face. And what you see is that if you only have one session, so one session of data for training, you're slightly above chance. But then when you start to have a lot of data, so 10 sessions and generalizing on 2 left out sessions, you almost reach 100% accuracy. Okay. So again, this really shows you that more data is better. The more you have data, the more your classifier should work on left out data. Okay, this seems obvious, but unfortunately, it's not always observed. And this is what I would like to show you here. So this is a work published by Gael Varocco in NeuroImage now 3 years ago. And what he showed is that it's basically a meta-analysis. And what he showed is that across many papers that are applying deep learning, machine learning, in the context of biomarkers, the performance that is reported in the papers tends to go down with the sample size. So let me just repeat this. The more people have access to data, the more the performance is going down. And for me, what it means and it demonstrates is that people are not doing properly the split between train, validation, and testing. Because if you're using the test data to optimize your parameter, the less data you have, the easier it is to get a good classification score. Okay, so here I think it demonstrates something which is a bit scary in our field, is that we need more data, but when we to predict well, but the numbers that we may find in actual papers can be quite misleading, because it's so easy to make mistakes and to be optimistic in the performance that you report. All right, now you will have a hands-on. Be happy to take a few questions from the audience. You will discover deep learning using M&E, PyTorch, using Hubert and Richard's notebook that they prepared for you. And I'll stop sharing and maybe answer your questions that you may have.